Here's a weird question. Why does evolution keep building crabs? Not just once. At least 5 separate times, completely unrelated crustaceans have looked at their lobster-shaped or shrimp-shaped bodies and slowly, over millions of years, reorganized themselves into the same flat, armored, claw-waving design. King crabs did it. Porcelain crabs did it. Even sponge crabs did it. And nobody can fully explain why. The phenomenon has a name. In 1916, a British zoologist named Lancelot Borradaile coined the term carcinization from the Greek word for crab to describe a pattern he kept seeing in crustacean lineages that were not technically crabs. They were turning into crabs anyway. They had different ancestors, different evolutionary starting points, different ecological niches, and they had all independently ended up with the same body plan: a wide flat carapace, a short tucked tail folded underneath, a pair of claws out front. The number of times this has happened in the fossil record keeps going up the more carefully people look, and the current count sits at a minimum of 5, and probably more. To understand why this matters, you have to back up to what evolution usually does. Most of the time, when 2 lineages share a feature, the simplest explanation is that they inherited it from a common ancestor. Eyes, spines, jaws, wings inside one group of insects. The trait shows up because it was passed down. Convergent evolution is the other story. It happens when 2 lineages, starting from different places, arrive at the same solution independently because the environment kept rewarding the same shape. Bats and birds both fly. They did not inherit flight from a shared flying ancestor. They each evolved wings on their own because air rewards wings. Carcinization is convergent evolution turned up to a strange level. It is not one body part. It is not one organ. It is a full reshaping of the entire body plan, repeated across crustacean groups that have been separated for tens of millions of years. The closest thing to a parallel in mammals would be if dolphins, seals, and otters had all separately ended up looking like the same fish. They sort of have, but with very different skeletons underneath. In crabs, the skeleton goes along with the makeover. The result really is, structurally, a crab. Start with the king crabs, because their story is the cleanest example of what this looks like in the lab. For most of the 20th century, biologists assumed king crabs were just very large crabs. They look like crabs, they walk like crabs, they are sold at fish markets as crabs. The body plan is unmistakable. But there was always something a little off in the details. King crabs have an asymmetrical abdomen tucked under their bodies that does not match the symmetrical tail of a true crab. They have a reduced last pair of walking legs that does not quite fit the standard crab leg count. The anatomy whispered that something else was going on. In 1992, a team led by Clifford Cunningham, Neil Blackstone, and Leo Buss published a paper in Nature using molecular data to settle the question. The DNA placed king crabs squarely inside the lineage of hermit crabs. Their closest relatives were not true crabs at all. They were the soft-bodied, shell-burrowing animals that you usually picture wandering around tide pools with a borrowed snail house on their backs. Somewhere in the deep past, a population of hermit crab ancestors stopped using shells, hardened up, flattened out, and reorganized into the king crab body plan. The Nature paper triggered a long argument that has only gotten clearer since. A 2024 genetic study out of the Chinese University of Hong Kong added another layer of evidence to the same conclusion. King crabs really did come from hermit crab ancestors. The crab shape is a destination they arrived at on their own. That is one lineage. Now multiply it. Porcelain crabs sit in a different crustacean family entirely. Their closest relatives are squat lobsters, which look like flat. The same thing happened. The body flattened, the tail tucked under, and the claws came forward. A porcelain crab and a true crab, lined up next to each other on a rock, look almost identical to a casual observer. They are, evolutionarily speaking, separated by tens of millions of years of independent history. They got to the same place from different starting points. Sponge crabs did it again, in their own way. So did hairy stone crabs. Coconut crabs, the giant land-dwelling animals that climb trees and crack open coconuts with their claws, are themselves descended from the same hermit crab adjacent lineage that produced king crabs, and they show a partial version of the crab body plan. Count of confirmed independent runs at the crab shape currently sits at 5 lineages, with a handful of partial cases on top of that. The natural question is why? Why does evolution keep walking up to the same blueprint? A 2021 paper in the journal Bioessays, led by Joanna Wolf and colleagues, tackled this head-on with a title that more or less summarized the whole field. The paper was called, in essence, How to Become a Crab, and it laid out the leading hypotheses for what is going on. The short version is that nobody has a single clean answer. The longer version is that the crab body plan probably solves several different problems at once. And those problems keep coming up in the kinds of environments crustaceans like to live in. One leading idea is mechanical. A flat, wide body is hard to flip over. It is hard to pick up cleanly with a beak or a claw. It sits low to the ground and presents a tough shield on top. For an animal that spends its time on a rocky seabed or in a tidal zone where predators are dropping in from above, a flat armored disc is a defensible shape. The tuck tail, which in earlier lineages was a long muscular swimming organ, becomes a folded plate that protects the soft underside without sticking out as a target. Several of the lineages that have carcinized also lost the ability to swim by tail flick along the way, which is the trade-off you would expect if the body was being optimized for sitting still and defending rather than darting away. A second idea is about locomotion. Sideways movement, the famous crab walk, is mechanically efficient for an animal with a wide flat body and legs splayed out from the sides. It is hard to do with a long lobster-shaped trunk. Lining up the body horizontally, with the legs to either side, gives you a stable platform for scuttling across uneven surfaces and squeezing into narrow crevices. The crab shape may just be the geometry that pays off best for a benthic crustacean that wants to live in cracks and on flat rock. If you are enjoying this little evolutionary puzzle as much as I am, hit subscribe. More stories like this on the channel every week. A third idea, less talked about, is about the digestive geometry. Some morphologists have pointed out that the carcinized body plan tucks all the soft organs inside a compact, well-protected cavity and reduces the surface area of vulnerable tissue. Whether that is a primary driver or a side effect is still debated, but it lines up with the broader pattern of the crab body being a shape that, once you arrive at it, is unusually hard to attack. Carcinization is not a one-way road. Evolution has also reversed the process. Decarcinization, the loss of the crab body plan after a lineage had already evolved it, has happened at least 7 separate times in the fossil and molecular record. Lineages that became crabs have then, later, unbecome crabs. They re-extend the tail, they lose the wide carapace. They go back to looking like something more lobster-shaped or shrimp-shaped or squat lobster-shaped. Whatever pressure was favoring the crab body in one environment, a different environment can favor undoing it. If the crab shape were a fundamental optimum, you would expect crabs to win once they arrived and stay crabs forever. Instead, the body plan keeps appearing and disappearing across deep time, like a fashion that keeps coming back. The crustacean body is, in a sense, plastic. It can flow into the crab shape under certain conditions and flow back out under others. The genes for the crab body plan are sitting in the broader crustacean toolkit, ready to be switched on when the environment rewards them and switched off when it does not. There is one more thread worth pulling. Comparative morphology work in the late 2010s has shown that the carcinized forms are not all built the same way under the carapace. King crabs, true crabs, and porcelain crabs differ in their internal anatomy, in the structure of their muscles, in the way their nervous systems are organized, and in the details of how the tail attaches and folds. From the outside, they look like the same animal. From the inside, they are 3 different animals wearing the same outfit. The crab shape is, in effect, a costume that several different organisms have learned to put on, and underneath each costume the actor is different. That is what makes carcinization so philosophically interesting. It is not really one thing. It is a pattern that nature keeps finding from several different starting points. Some kind of selective gravity, in the kinds of environments crustaceans like, keeps pulling unrelated lineages toward the same external form. The genes underneath are different. The muscles underneath are different. The evolutionary histories are different. But the shape at the surface, the wide flat carapace, the tucked tail, the claws out front, is the same. We tend to think of evolution as a process that explores. Different lineages wander off in different directions, find different niches, and produce different animals. Most of the time, that is exactly what happens. Carcinization is one of the few examples where evolution looks less like exploration and more like a series of independent expeditions all stumbling onto the same destination. Tens of millions of years apart, from different starting points, by different routes, and ending at the same crab. Borradaile, when he gave the phenomenon its name in 1916, almost certainly did not know how often it had happened. He was looking at a few cases and noticing a pattern. The molecular and genetic tools that have unspooled the real picture only arrived in the last few decades. The current count of independent carcinization events keeps going up as more lineages